Hi, everybody, and welcome back. This is chapter 12. Here we're going to be talking about antimicrobial treatment. In case you haven't figured out, we're talking about antibiotics in this chapter here. So let's get started. Here are our learning outcomes for this chapter. It looks like there's a lot more, but I think it's just more words involved, but it might be more. But this is the stuff you should be able to answer. And if you guys want to come back and test yourself over this stuff afterwards, I would recommend. So antimicrobial chemotherapy. Chemotherapy, by the way, it just means treating any disease with a drug. So treating something with a drug, that's chemotherapy, using chemicals as therapy. So antimicrobial chemotherapy, administering drugs to a person, infected person that destroys the infective agent without harming the host's cells. That's the goal, right? But the perfect drug doesn't exist. And we'll talk a little bit about how come. So we want an ideal antimicrobial drug to be toxic to the microbe, but completely safe for the host. We want to be microbicidal rather than microbistatic, which you'll learn about soon. But that means microbicidal means killing the organism instead of just stopping it from growing. Soluble in body fluids, right? So that whatever, that makes sense. Remains potent long enough to act, is not broken down or excreted prematurely. Of course, it stays in the body long enough that it actually acts on the microorganism. You don't have to keep taking and taking and taking and taking. And then slow or non-existent antimicrobial resistance. Hopefully non-existent. That's the hope. We also want something that complements or assists activities of the host's actual defenses, maybe helping with maintaining immunity afterwards. Remaining active in the tissues and the body fluids. We already kind of talked about that. Readily delivered to the site of infection. And so it doesn't have to, wait too long to get to that area or be hard for that antibiotic to get to that area because it depends so highly on proper blood flow. Because, you might not be able to have proper blood flow to certain areas where you have infection. So, and we also want something that doesn't disrupt the host's health with allergies or something. So prophylaxis is using a drug to prevent infection. So that's like when you go travel to areas that have malaria and you take the drug just to keep from getting the malaria. That's an actual drug that treats malaria that you're taking. And so you're taking that treatment drug as a means to actually prevent you from getting it at all. So that if you were exposed to that organism, you don't get it. So that's prophylaxis. It's just like prophylaxis whenever we're talking about birth control. Birth control, in case you didn't know that, is prophylaxis. Antimicrobial chemotherapy, the use of drugs to control infection. Antimicrobials, anything where we're going to be targeting any microbe, regardless of the type. And then antibiotics, now we use this quite simply to refer to targeting bacteria and not other ones, okay? We're now talking about targeting bacteria with these. That's important to remember, by the way. Antibiotics, even though it says biotics, and the other ones are like antiviral, antifungal, antiprotozoa, they're obvious. Antibiotic, that's only bacteria. Please remember that. Semi-synthetic drugs, synthetic drugs. Semi-synthetic drugs are drugs that are based off of natural sources. Synthetic drugs are going to be created entirely in the laboratory setting. Narrow spectrum versus broad spectrum kind of speaks for themselves, I feel like. So the drug's going to affect mainly one kind of bacterium for the narrow, whereas broad spectrum, a large swath of different kinds of bacteria, maybe all gram-positive and gram-negative, whatever. Antibiotics are common products naturally of bacteria and fungi to inhibit growth of things that are going to be trying to live in their space, right? Competitors. So they would produce it for that reason. That's why the bread, moldy bread, would kill bacteria. It can be used to treat infections. That mold was producing antibacterials because, doesn't want the bacteria to grow. Keep it away. This is my food source. That's what it was saying. Yeah. So it is an advantage that can be passed on from generation to generation. Yeah, often the times, especially whenever you're being subjected to antibiotic treatment, you're selecting for the organisms that are going to be resistant, right? Semi-synthetic drugs, this would, maybe you had natural penicillin at first, then the organism had enzymes that could break it down. So now we've developed some with side chains that won't be broken down as easily by enzymes. That's an example of a designer drug. Then we have synthetic drugs, like designer drugs that could be manufactured straight up, like from scratch. Right. Manufactured compounds have largely proved unsuccessful in permeating the membrane because we don't have a wide enough understanding of how to get something into a cell. So that's why a lot of the stuff is based off of the structures of existing drugs. Okay, so if you're starting treatment with these drugs, you want to identify the microorganism. You want to know the degree of its susceptibility to various drugs, so you can figure that out. If you can't be told, then you can do tests to figure it out. And then the overall condition of the patient, of course. So identify the agent, do all of your testing that we talked about in chapter 13. Do all your testing that you can to try to figure out the causative agent for whatever this disease is. And then you'll want to do that before any antimicrobial drug is given. Before any antimicrobial drug is given, you're listening to me, right? Future physician assistants who would be prescribing these things. Yes, before. You want to know what it is before, as much as you can, right? There are certain diseases that you might get, things like meningitis, that you will want to start treatment with something before you even know what that organism is. You know what it most likely is, though. So that's a difference there. It's not like you're just trying to target all bacteria. The problem with this is you don't want to just throw everything at something with a shotgun mentality, because then you are being a proponent, or you're taking part in the whole problem of developing antibiotic resistance. Yes, you are. That is how that works. That is why you're supposed to take your antibiotics to completion. If you start feeling better and you stop taking them, there are more likely to be microorganisms, bacteria in your system that are going to be resistant to that antibiotic as a result of you stopping them, because you didn't kill them all the way. So do not do that, and do not just prescribe antibiotics for no reason. Direct examination of body fluids, sputum, or stool is a useful means of determining what the organism is as well. So we can use Kirby-Bauer technique. We learned about this in lab, right? You plate your lawn of bacteria, you pop some antibiotic discs on there at certain concentrations, and then you see the effectiveness after you grow it for 24 hours or so, to determine how susceptible your microorganism is to each antibiotic at each concentration. The useful way of determining the antimicrobial sensitivity of that organism, that is called an antibiogram of that organism to that antibiotic, right? Antibiogram, determining this answer for a lot of different antibiotics for your organism of interest, your patient's sample, right? So you got a throat swab and you swabbed on it, and then you stamped your little antibiotic discs on there, and then you grow it, and then you come back the next day and you find out that here we have every time. All right, here we have your antibiotic, your organism is susceptible, right? It's being controlled. It can't grow here because of it. Whereas this guy here, clearly your microbe is resistant completely to that. You would not want to give this to your patient. You would not want to give that to your patient either. That's vancomycin. That patient doesn't get this, okay? So we would want to give it one of these other ones that it is more susceptible to, hopefully. Like tetracycline, I think, is one of these. I don't know which one it is because I don't know the little abbreviations that well. Sorry, I was trying to stare at it and figure it out. Okay, so there's also this other test called the E-test. It's the same exact concept. On the E-test, basically it has increasing levels of antibiotic on there for whatever antibiotic you're looking at. And so I guess I'm assuming this is azithromycin, but I don't know. And then the concentration increases down here. And so that's why we would be getting more growth out here. So these are the zones of inhibition, ZOI, remember? Okay. So the minimum inhibitory concentration is the smallest concentration that the drug that you can use that will visibly inhibit growth on these tests. All right, so we can use a tube dilution test as well. It's just a dilution of the actual drug dosage to a point where the minimum concentration can be found that would be useful to treat a patient, right? So we have machinery apparently now that can do this in certain hospital settings. Sorry, my brain, you know how it is. So this is our positive control. This is what the growth looks like. So if we are introducing and we're doing dilutions, it looks like right here is the point where, okay, now we have a problem. Now this is where this will become, not say we have a problem. That's not really what I mean. So this is what growth looks like. So these we're able to growth. These are too diluted. This is the concentration where it starts to work against the bacteria. So, bacteria have a problem is what I was trying to say, but it's confusing. So starts working here. So this is more diluted drug. This is more concentrated drug. So this one, we had to use the highest amount of drug. This one, we could do this one and this one and whatever for each of these drugs, determine the actual visible level where we would say definitely killing everybody in this concentration. So that could tell you what concentration of potentially harmful drugs we can use to treat your patient. Okay, so we want to find our suitable drug, but what if you treat your patient and it doesn't work and you do these tests that you just did that I just showed you? So that could mean that the drug is unable to diffuse into that body compartment for whatever reason, not getting enough blood flow, whatever it is. Can't cross the blood-brain barrier, whatever. It could be that the drug, there are resistant microbes in the infection that whenever you did your Kirby-Bauer test, didn't get chosen, wasn't one of the chosen ones to grow on the plate, or didn't actually see that antibiotic on the plate. So that's a possibility, unfortunately, if you didn't pick up the ones that are resistant. This isn't like lab, right? It's not a perfect scenario. There might only be 1 or 2 bacteria that are resistant. And then now, because you treated your patient with it, now they are overgrowing because they're the ones that are surviving. And then it could be also that you have a infection caused by more than one pathogen. So it's a mixed bacterial population, and some of those are resistant. So that could be possible, and you only picked up one for your testing. So the therapeutic index is the ratio of the dose of the drug that is toxic to humans. How much can you take of this medication before it starts being bad for you, versus its effective dose? So how much of that medication do you need to take in order to kill the bacteria? So the ratio of those two, the closer that those are to one another, we're towards a danger zone. So that's working on a potential for a toxic reaction to that drug. Therapeutic index of 1.1, that's a closer value, is a riskier choice than a therapeutic index of 10, because it's farther from 1. And the farther you get from 1, the better the scenario. When drugs have similar MICs, which is that minimum inhibitory concentration, so the minimum amount you need for killing, when drugs have similar values for that, the drug with the highest therapeutic index will have the widest margin of safety, and that'll be the one that we'll choose. So we'll base it on a lot of these factors, right? So we're not just saying, oh, okay, it grew on Kirby-Bauer, or it didn't grow on Kirby-Bauer, and that's the drug that we're giving. So it's more complicated than that, right? So you have to consider the dangerous aspects of these drugs. The therapeutic window is the range of the blood level of the drug in which it's producing the desired effect without being dangerous for the patient, being toxic for the patient. Yes, if you have ever seen any patients that were on these super strong antibiotics, you will see that there are definitely negative side effects to these. We also have to consider the patient's history. So this is a particularly toxic drug and you have a particularly weak patient, you might want to rethink your decision. So if they have underlying liver or kidney disease, they might not be able to process the drug properly, to get rid of it. They might have pre-existing medical conditions. They could have allergies to certain ones. Are they elderly? Are they pregnant? Do they have certain autoimmune diseases? anything like that, babies. So it's not super straightforward even after that. There are synergistic and antagonistic effects of drugs, basically talking about effects of drugs on one another, depending on what other drugs you could be on, as well as antimicrobials affecting your actual human biome, which is unavoidable, unfortunately, but that will happen. Not a maybe, it will happen, but to what degree is what we want to know. So selective toxicity is a concept where we are, that's one you're definitely going to be tested on, by the way. Selective toxicity is referring to the fact that you want your antimicrobial drugs to kill or inhibit microbial cells without causing damage to the host. So that whole concept of kill the bad guy, safe to the host. That's what selective toxicity is. The best drugs in current use block the actions of synthesis of molecules in microorganisms, but not vertebrate cells. So we're vertebrates. And drugs with excellent selective toxicity might include the penicillins because they attack the synthesis of the cell wall of organisms, which is something we don't have in our cells, right? So metabolic targets of chemotherapeutic agents. We could inhibit cell wall synthesis. We could inhibit cell membrane function, nucleic acid structure and function, folic acid synthesis, and protein synthesis. So here is a diagram showing all of the drugs that fall under these various categories, things that inhibit the cell wall synthesis, things that inhibit folic acid synthesis, and etc. I'm not going to go through this now because we're going to go through them, of course, slide by slide, but that's a great reference point to come back to. All right, at least 5% of all persons taking an antimicrobial drug experience some type of serious adverse reaction. This could be direct damage to tissues through toxicity. This includes like tetracycline causes a gray line on your teeth if you keep taking it. This could be development of allergic reactions, or it could be disruption in the balance of your normal microbiota causing you issues as a result. That's usually the case with development of pseudomembranous colitis. So C. diff infection can develop as a result of taking antibiotics to treat other microorganisms. So toxicity to human organs, we have the toxicity to liver, whatever, a lot of human organs that it could affect. You can see it yourself. Drugs act as antigens, our body sees it as foreign. We respond to it accordingly, and we can develop allergic responses as a result. Penicillin and sulfa drugs are the most common for this situation. After antimicrobial, sorry, I don't know why I didn't see that. After an antimicrobial destroys beneficial resident species, others will overgrow. So killing all of your bacteria in your normal body, and then now your yeast in your genitourinary tract, so your vagina and all of that, that's going to not be killed by an antibiotic because it's not a bacterium, by the way, right? So yeast is a fungi. So Candida albicans is the yeast it's normal. You should have this as part of your normal biota in your female reproductive tract. But we take antibiotics, we kill off all of our bacteria, and now the yeast can overgrow, and now we have a yeast infection. So that is exactly the same idea as what we get with C. diff, which is Clostridioides difficile or Clostridium difficile. They've changed the name recently, but anyways, same idea there. So it's a bad thing. That's called superinfection. So here is just showing spectrum of antibiotics. You can see here the tetracyclines have quite broad spectrum, whereas the polymyxins have a very narrow spectrum of what kind of bacteria they can actually treat. So the first set that we have are our cell wall drugs. These ones are going to be our penicillins, including our penicillins. They all are going to have this group called a beta-lactam ring. So this is the beta-lactam ring, looks like a square in this picture. And that is a defining characteristic of these drugs. So we have the beta-lactam ring, the thiazolidine ring, and a variable side chain of some kind. I don't need you to worry too much about that, but definitely be aware of that beta-lactam. That's very important. We can't synthesize versions of these in the laboratory. So then we also have the cephalosporins. They also have a beta-lactam ring. and they were isolated from bacteria. And then we have similar mode of action as a result. And then the carbapenem, guys, that we reserve these in hospitals when other drugs are not working well. Other drugs that can target the cell wall, bacitracin, isoniazid, and vancomycin, they are not going to have beta-lactam rings on those guys. Okay, they're different. Yeah, so this is just talking about more characteristics of those last three. So the aminoglycoside drugs, these are going to affect protein synthesis. They're usually amino sugars and a 6-carbon ring of some kind. These are products of actinomyces. So some organisms that fall under this, streptomyces and micromonospora. I'm not going to ask you that. But anyways, I told you. So aminoglycosides are broad spectrum typically. So this is the structure of them. There you go. That's streptomycin. Here are some of our aminoglycosides that and how they affect the function of a ribosome. Most of these drugs are actually going to be involved in inhibiting the movement or actual function of the ribosome and how it works in the cell. So I'm not going to go through the details of these right now because I'm not going to test you on the details of how these work, but I would be aware that they affect the ribosomes. I would be aware of aminoglycosides. I would be aware of glycylcyclines and tetracyclines and erythromycin. If you haven't heard of these terms, I don't think you've been working in the medical industry. I don't believe it. So tetracyclines were one of those categories, right? They are derived from streptomyces. Cool. Natural parent compound and synthetic derivatives of this exist. So there's several kinds of this, but they're going to block the ribosome and therefore protein synthesis. Then we have the macrolide antibiotics. They also associate with protein synthesis, stopping protein synthesis. I don't really know what else to say other than they are macrolides. This will include clarithromycin and azithromycin. So that's one to be aware of. Azithromycin is a macrolide. It's usually useful for middle ear infections, as well as respiratory and skin infections. So azithromycin is probably the one that you guys are more familiar with. I don't know clarithromycin very well. Then we have our sulfa drugs. This even includes a silver sulfadiazine, which is for burns, right? So our sulfa drugs, if it has a sulfur aspect to it, it's probably going to be involved in folic acid synthesis. Trimethoprim is one of these. It's often given in combination with sulfamethoxazole, which is another one of these. But anyways, they're sulfa drugs. People can be allergic to these, usually allergic to the whole group, right? These are going to mimic one of the intermediates of folic acid synthesis. So they need folic acid for a lot of things, bacteria do. Typically for like DNA and RNA and amino acid production. So that's a big deal, right? So if we can inhibit that, it really puts a damper on their party. So what we do is we mimic that with our sulfa drugs and it binds into the enzyme. That's a competitive inhibition there, right? Remember that, competitive inhibition. Next, we have the ones that affect DNA and RNA. These are the fluoroquinolones. They are high potency, broad spectrum, absorbed in the intestine, and they have serious side effects like seizures. Because if we are affecting DNA is pretty universal. Sorry to break it to you, whether you're talking about bacteria or eukaryotes, they're pretty similar. So this is just talking about some of the fluoroquinolones. I would be aware of ciprofloxacin, and ofloxacin. But anyways, the floxacin, that's a fluoroquinolone. Anything floxacin, just think of it that way. And then rifampin is another one that falls under the category of ansamycin that are associated with DNA and RNA inhibition, but I'm not too concerned about that. So the polymyxins are going to be associated with targeting the cell membranes of organisms. These are developed from bacteria Bacillus polymyxa. Cool. They are pretty narrow spectrum. They have a fatty acid component that allows them to act almost like a detergent and get in between the phospholipids of the membrane and break it down. This is toxic to our kidneys. So we don't typically use this as like a medication that you like infuse or swallow or something like this. We use this as a skin ointment. This is included in Neosporin. Just FYI. Then there's daptomycin, which is related to this, which is another lipopeptide, so lipo being a fatty acid group. Okay, drugs that can affect biofilms. You want to have something that can penetrate the whole biofilm and treat all of the organisms involved in the biofilm typically. So you want things that can interrupt the quorum sensing pathways. That's the ability of the bacteria that are in the actual biofilm to communicate with one another. And then we will also treat with daptomycin because most of the things are going to have membranes. It's just, typically have a membrane. And then we have pre-treatment if you are going to be receiving like something like a pacemaker that could develop a biofilm on it, and pre-treat with a medication so that you don't develop the biofilm. All right, then we have fungal infections. The fungi are eukaryotes, and so that's going to not be antibiotics, right? So it's going to be antifungals. Most antibacterial drugs act on bacteria, and they would not be useful on fungi. So because we are also eukaryotes, this is where the problems start, right? So our cells are more similar to their cells. So treating us can cause problems with the medications. So here are some ones you might be aware of. We have our macrolide polyenes. This includes amphotericin B. That's a big antifungal. You should be aware of that one. This will treat skin mucous membrane lesions caused by Candida albicans. It also includes Cryptococcus meningitis for Cryptococcus meningitis, right? So that would be pretty much speak to you as far as that. But anyways, those are pretty versatile drugs, the amphotericin B drugs. I actually had to use a sinus spray that was made with this. I had to go to an apothecary to have it made. But I had it kept in the fridge, and then I had to shake it. Anyways, azoles in general, but not always. So we do have some antibiotics that end in azole, right? But in general, otherwise, the azoles are going to be antifungals. So this includes ketoconazole and fluconazole and miconazole. This miconazole, this is what you get in like Monistat. So if you don't know what that is, then you're probably not a lady. So, because most women probably have some sort of experience with this. And then the other ones are pretty common for treatment of some of these other things like aspergillosis, which is a very common, aspergillum is a common fungus in the environment that people can get as, an illness, whatever. So, and they'll often give it as a pill if you have a yeast infection. Yeah, cool. I don't care about the others. So then next we have the anti-malaria drugs. These are usually all based on quinine. Quinine is was for a long time like the major go-to treatment for malaria. Now we use chloroquine and primaquine. They are less toxic to humans, but still have negative side effects associated with them. But it was way worse with quinine, believe me. So, yeah, it doesn't treat all the species of Plasmodium, unfortunately, but we can give this as a prophylactic treatment if you are traveling somewhere with malaria. So we usually combine it with other drugs if we are actually treating you, if you know you have malaria. All right, protozoa and the helminths. So these are, so protozoa, that's going to be, that includes like malaria, by the way, but then we're going to talk about other protozoa. And then the anti-helminth drug therapies. So other protozoas could be like trichomonas. That's a pretty common one. Yeah, it's a sexually transmitted disease. Anyways, metronidazole, very common treatment for amoeba type organisms and other protozoa. It's also useful for anaerobic bacteria, by the way. The anti-helminths, those are going to be like a lot of these, most of you guys will be hearing of as far as like veterinary studies, because we don't deal with helminth infections that much in people. So this will be like heartworm preventatives and stuff. Most commonly ivermectin and praziquantel. Those are the most commonly used ones. And even for human helminth infections, that would be the case. So with antiviral medications, we want to keep the virus from getting into the cell. And then if we can't keep it from getting into the cell, we want to block replication and transmission and translation. So the building of the building blocks of the virus. And then we also want to prevent the maturation of viral particles if we can't do the other two before that. So this is just going through some different medications that are used as antivirals. So here we have blocking HIV infection by preventing binding of viral receptors to the cell receptor, so keeping it from getting in. So this is, by the way, receptor fusion and uncoating, keeping it from getting in, right? Amantadine. This is going to be Relenza or Tamiflu, so flu treatment here. These are going to stop the action of the influenza neuraminidase. If you've ever heard of H1N1 or something like that, H5N1, whatever it is, H is hemagglutinin, N is neuraminidase. And this is used by the virus to essentially break its way into our cells. And so if we can keep it from working, we can keep it from getting in. Next, we have Okay, these are the guys that are going to inhibit nucleic acid synthesis of the virus, right? So this is going to include remdesivir and ribavirin. Ribavirin is probably the more common one that you've heard of. I have not heard of remdesivir, but I guess they use this for COVID. I guess it's because I haven't really been to the hospital since COVID, but it mimics the typical nucleotides that are involved, the ATCG parts. It mimics those, and that stops it from being able to make RNA. So that's what that one does. This one has a similar function, but we use this for Hep C and other diseases. All right, we have our nucleic acid synthesis that are for other viruses, of course, HIV, some more. So HIV is unique because it involves reverse transcriptase. It takes RNA and turns it into DNA and then inserts it into our own genome, right? So we can stop that because it's something that isn't found in our cells normally. So we can block that and then hopefully block HIV production. Unfortunately, these guys do mutate quite quickly. Then we have another non-nucleotide analog, reverse transcriptase inhibitor. So reverse transcriptase inhibitor here, preventing that reverse transcriptase from making the DNA from the RNA so that it can't actually do that part and allow for the HIV to insert itself into our genomes. Now we have viral assembly and release drugs. This would be idinavir and sequinavir. These are protease inhibitors. These typically cut up the proteins that are made as a product of reading the mRNA for HIV, and so it cuts it up into usable pieces. And if it can't cut it up into usable pieces, it can't use the pieces, and therefore it's not going to be a active virus. All right, so drug resistance, how does that develop? It's usually an adaptive response for microorganisms. They can tolerate a small amount of drug that would typically be inhibitory. So any sort of genetic mutation could potentially kind of cause that if it's in the right place, right? So this is usually a genetic versatility and adaptability of microbial populations in general. They just multiply so fast that eventually there is likely to be some sort of a mutation that will be advantageous in that way. So intrinsic resistance, that means that if you produce penicillin, you will be resistant to penicillin. Like you have to be. So that's an intrinsic resistance. If you got a plasmid that encodes an enzyme that helps you break down penicillin, and you got that via conjugation through the sex pili, then that would be acquired because you got it from other bacteria, right? So you got it, you were previously would have been sensitive to it, and now you are resistant. So spontaneous mutation is one way that you can get this, right? You're just multiplying so many times, you're likely to have eventually the mutation that would be advantageous in this way, but it's a very minimal chance. It's an even smaller chance than that, then the mutation will confer actual drug resistance. But since there are so many of them multiplying so much, it is actually likely to occur at some point. So when we're talking about horizontal transfer, that could be conjugation, transformation, or transduction. We talked about this previously. Please tell me that you remember what those are. And then resistance factors are plasmids that have some sort of drug resistant gene associated with them that can be transferred by these methods. Transposable drug resistant sequences are going to be on transposons. These can jump from plasmids to the chromosome and back and forth, and also be transferred via the conjugation, transformation, or transduction. By one of those methods, and then become, transposons once it gets to the new place, then jumps into their chromosome, and so on and so forth. So that can happen. So that's transposable drug resistance. So we have resistance factors, that's the plasmids. Transposable drug resistance, those are transposons, they're jumping genes. So rapid proliferation of drug resistant species, the gene transfers are extremely frequent in nature. So if you have one guy that has the gene that protects it, it's more likely that you're going to have more than one eventually, like they're going to multiply pretty free and quickly, right? So gene transfers are very frequent in nature and just naturally. And so that is more likely to transfer the drug resistance as well. And genes from unrelated bacteria, viruses, and other organisms live in the body's natural, normal biota and environment as well. So they can be exposed that way. All right, so mechanisms of drug resistance. We have 5 different ways that we can deal with this. So the first one are new enzymes can be synthesized so that can break down the drug. This one, it says to us that this will only happen whenever new genes are acquired somehow. We also have permeability or uptake of the drug into bacterium being decreased. Basically, this means that they are changing the locks and the drug no longer has the key that works. So the drug can't get in. So that's this one here. They say that one usually occurs via a mutation. And then number 3, our drug will be immediately eliminated. That's this one here. That just means that we're going to take the drug into the cell and then immediately shuttle it right back out. So just don't let it sit around in there enough. So this is usually also one that involves acquisition of new genes. So yes, that's number 3. So then we have 2 more. Number 4 and number 5. So number 4, binding sites for drug are decreased in number or affinity. So, drug doesn't bind the drug very well, or I don't know, doesn't have very many spots where it can fit anymore. So in this case, we have our drug that normally binds here, and now we've changed it a little bit and now can't stop the ribosome from working anymore. So it has changed its ribosome shape essentially to respond to whatever that drug was. This is usually a mutation or new genes, so one or the other. And then number 5, an affected metabolic pathway is shut down or an alternative pathway is used. This is typically what we're talking about with like folate synthesis. So this is for, and this is okay. Okay, so normally we go straight on through here, and normally the drug acts about here. But instead, now we've developed a new way of, okay, no, instead we're going to turn it into this other molecule instead, and then this one goes into that molecule instead, and then we'll get to the product this way by this chemical reaction instead of the original one. So we're going to bypass your stupid little drug and get around it. So that's the idea of how drug resistance will work. So how does all this happen? I've kind of already brought this up. If you guys don't really understand this, I don't know really what to tell you. You have a few bacteria that are in a population that might just be chilling, that just happen to have these mutations, or plasmids that could confer resistance to a drug of some kind. Now you come in and you treat your patient with vancomycin, and the 2 bacteria of all of them that happen to be resistant to vancomycin will now overgrow and make up most of that population. And any of them that happen to survive that didn't have the resistance, they can transfer it horizontally by conjugation, transformation, or transduction, and transfer those genes that way to be protective for those organisms. All right, 75% of antimicrobial prescriptions. Read that again, folks, because if you are a PA student and you will be actually writing prescriptions, or if you are a nurse that will be going into nurse practitioner, and you'll be writing your prescriptions. 75% of antimicrobial prescriptions are for throat, sinus, lung, and respiratory infections. Most of those are going to be viral, honey. Most of them are viral. Do you treat viral infections with antibiotics? No. So don't be doing that. We often use the shotgun approach to just treat like, oh, you have a sore throat, here's 90 antibiotics and go home and you'll be fine. And you will be, but you'll also be harboring antibiotic resistant bacteria. So the next time that you get something, you could potentially be resistant. So think about that. So tons of excess antimicrobial drugs are transported to other countries. They'll become resistant, and then that can be transferred eventually back to us. Hospitals often have weakened patients, and the workforce doesn't adhere to universal precautions, right? We know that's the case, not always intentionally, right? But sometimes it just happens by accident. So this has led to penicillin resistance. About 100% of Staph aureus is resistant in some way to some kind of antibiotic. This isn't MRSA. So this isn't multiply resistant Staphylococcus aureus. This is, we're talking here about just one kind of antibiotic at first. So this is not quite as bad, but still bad, right? It's not good. So 100% of it is resistant. So 80% of antibiotics in the United States are given to livestock. This supposedly decreases infections and improves animal health and size, whatever, but these pathogens are developing resistance to these drugs and can jump from the animals to people and cause resistance in us. Right, there's some things that are becoming so antibiotic resistant, it's a very serious concern. We have the urgent threats, which are the worst, the serious threats, and then the concerning threats that are less serious. Our urgent threats, these are the worst ones. Carbapenem resistant Acinetobacter. I don't know much about these, but I know that carbapenem is like a last line defense antibiotic. So that's a bad deal. And then we have Candida auris, which is a fungus, Clostridioides difficile. So that's C. diff. We have C. diff becoming resistant to certain antibiotics and whatever. And so one of the biggest issues right now is actually drug resistant. When I say drug resistant, I'm talking about like to almost every single drug ever in the history of ever. This is gonorrhea. It's no joke, man. That's a serious, serious issue. And then we have a lot of these other bacteria and diseases. You know, you can look over these on your own time, but we do know that we have a lot of streptococcus and staphylococcus out there that are drug resistant. So those are common knowledge, I feel like. So in the past, whenever we want to find new drugs, we would target, known bacterial targets and then custom design drugs to aim for those known targets. But now we have a new way of approaching things that's giving us a little bit more variety in the drug choices that we could potentially come up with. So we have nanomaterials, antisense RNAs that could block the ability of these things to work, CRISPR technology to cut into certain sequences of the DNA. We have bacteriophages that can affect specific bacteria that we can, target to, be useful for us that way. And antibiotics that will be targeting critical gram-negative outer membrane proteins that are associated with those gram-negative and whatever, and they shouldn't be in the areas where they are and whatever. We have bacteriophages that have been incorporated into bandages for wounds that have been extremely effective as far as stopping bacterial infections there. So ones that are typically associated with those wound infections, bacteriophages against those bacteria. So probiotics, they help nature along. They're going to replace any of our lost microbes that we lost from taking antibiotics, as well as just supplementing and helping out our natural biota. They're very safe, they're very effective, and there's no reason why you shouldn't be incorporating them into your diet. Prebiotics are nutritional aids that help with growing those probiotics. So that's pretty useful as well. So fructans encourage the growth of bifidobacterium in the large intestine and discourage the growth of pathogens. So they'd be very useful as well. So always have your probiotics with your prebiotics, always. Then we have fecal transplants. This is kind of what it sounds like, but I don't know how to tell you about it, how it's not a bad thing. You have C. diff. We've depleted your entire, flora with all these antibiotics. and C. diff has overgrown, and now you have, pseudomembranous colitis, and we need to help you out here. So what can happen is we can eliminate issues with that by introducing new healthy microbiome, usually via colonoscopy into the patient from typically somebody who has a healthy biome that lives close to that patient, and then replacing their biome with theirs. And then it actually works quite well. So yeah, it's useful. So that's the end of this chapter. So our end of the chapter question: describe a realistic scenario that would involve the use of the Kirby-Bauer assay. That should be an easy one for you guys. And I'm going to go, and I'll see you guys next time. All right.